Bismillahirrahmanirrahim. So in continuation of our previous lecture on Mendelian genetics, in this lecture, we are going to learn about dihybrid cross, in which we are going to simultaneously follow up two different traits. For example, round seed shape and green color, wrinkled seed shape and yellow color. You may remember in monohybrid crosses, yellow was dominant over green and round was dominant over wrinkled, and we got 3:1, 3 rounds and 1 wrinkled, 3 3 yellows and 1 green, F2 phenotypic ratios. The next question he tackled was if two independent traits like color of the seed and shape of the seed, when you follow them simultaneously from parents to offsprings, so you cross round, green, yellow, wrinkled, what happens in F1? And when you self-cross F1, what happens in F2? Towards the end of the lecture, we'll emphasize on rediscovery of the Mendel and learn about how people realize that what Mendel discovered actually made sense, and how these dihybrid crosses or following two different traits, they fit in our latest and modern understanding of chromosomal theory of inheritance, that genes reside on the chromosomes, and in particular, inheritance through meiosis. So I hope you will enjoy this lecture, and this is also done for non-experts, for people who have never taken biology. So let's continue. So what Mendel did, he studied these 7 different traits. So round, spherical versus wrinkled, yellow versus green. Yellow was dominant and green was recessive. If you look at the ratios, F2 ratios on the top of this table, so F2 generation, the numbers for dominant in F2 and recessive, and then the ratio, the total number, and then the ratio 3:1. Similarly, the purple is dominant over white, inflated pods with constricted pods, and so on. What we just saw, that is referred to as monohybrid. What was monohybrid? We crossed round with wrinkled, or purple with white, or yellow with green. When you are focusing, when you are analyzing a single trait, that is what we call mono. Mono means single, one. But Mendel came up with an idea that what will happen if I start using 2 different traits? What if I mix them? In one parent I have round and then yellow, and the other one is wrinkled and green, both traits in the same parent. That is called dihybrid cross. Okay, when we are going to analyze 2 different traits. Before we move on, another very important discovery Mendel made on the basis of his experiments was that there is no blending. There is no blending. Why we are coming up with blending here? Because before Mendel, there was an idea which was called blending theory of inheritance. blending theory, which means you cross round with wrinkled, you know, F1 will show you semi-round, semi-wrinkled. But in a case of Mendel, he did not find any blending. It was completely dominant. Round was completely dominant. Yellow was completely dominant over green. Tallness was completely dominant over dwarf. So it was not that you cross tall with dwarf and in F1 you get intermediate. No. So there was no blending. So that's why in this slide you see no irreversible blending. So the blending theory was basically rejected by Mendel's experiments. What Mendel did next was the dihybrid cross. He said, okay, I know that whenever I do monohybrid cross round with wrinkled, F1 is all rounds. And when I self-cross F1, I get 3:1 ratio round versus wrinkled. Similarly, when I do a monohybrid cross and I use yellow with green, all the F1 was yellow. And when I self-cross F1 yellow, I get 3:1 yellow green. And he knew that, you know, since yellow is dominant, he used capital Y, green is recessive, small y, F1 was this one. When you self-cross F1, these yellows, which are heterozygous, you get three yellows, one green. That phenotypic ratio, 3:1. And of course, when you go to F3, take randomly 100, you get 1:2:1. This was what? Genotypic ratio. Genotypic, because this is based on the genotype. Phenotype was simply based on the appearance. So he said, okay, I have studied 7 different traits. All 7 showed me complete dominance. All showed me 3:1 F1, 3:1 F2 phenotypic ratio, and 1:2:1 genotypic ratio. But what will happen if I analyze 2 traits at the same time? In the experiment he did, he said, okay, I'm going to take, parents are going to be yellow wrinkled, yellow wrinkled, and you can give the genotype of yellow will be, we know from our monohybrid cross that yellow is dominant, so double Y, and wrinkled is recessive, small r, small r. Green is recessive, small y, round is dominant, capital R, capital R. So based on the monohybrid cross, what are the facts we already know? I already explained you, round is dominant, so 3:1 F2 ratio you have, and same is the case for green, yellow versus green, 3:1 ratio, and there were genotypic ratio 1:2:1 for round versus wrinkled, and 1:2:1 genotypic ratio yellow versus green in the F2. However, the question was, these are facts when we have monohybrid cross. Do these facts hold true when we have a dihybrid cross And dihybrid means now we are analyzing two different traits in the same genetic cross. So the dihybrid cross is, you have yellow wrinkled, green round. F1 will be, what will be the phenotype of F1? What is the phenotype of F1?
A yellow round.
Yes. And why we have this genotype of F1? How is it possible?
Sir, because they get one from each like parent. So like one has two capital Y and it gets one of those and then one small y from the other one.
Yeah. So the possibility of this one contributing to this one, knowing the law of segregation, it can be capital Y with this small r, or this capital Y with this small r, this capital Y with this small r, and this capital Y with this small. So 4 combinations. And same is the case here. So this small y with this capital R, this small y with this capital R, this small y with this one, and this small y with this. But all of them will look like capital small r capital Y, small y capital R, and when they will mate, the F1, all the F1 will look like this, no matter which of these gametes meet. So the phenotype will be yellow and round. So F1 will be yellow round. When you self-cross F1, which means this one with this one, can you make the contribution of this parent to the next? Can someone tell me how many combinations will be there?
So 16?
16. But first tell me what this parent will contribute.
So capital Y, capital R, capital Y, small y, capital R, and small y, small r.
Very good. So 1, 2, 3, and 4. Okay. So what Mendel did, they said, okay, we go to now next generation, and he got 16 combinations. He just calculated their phenotypes, just phenotypes. And look at the phenotypes. He got round yellow, 315, round green, 108, wrinkled yellow, 101, wrinkled green, 32. You can simply draw a checkerboard here like we draw for the monohybrid. So on top, you can write here, all these gametes. On this side as well, capital Y, small r, small y, capital R, small y, small r. And if you write on this side as well, capital Y, small r, small y, capital R, small y. So you will get these combinations, and based on phenotype, you got this. Now what Mendel said, he said, okay, let's see if the underlying, these ones, the facts from monohybrid, whether these ratios, round versus wrinkled or yellow versus green, they are conserved or no. And what Mendel did, yeah, here. What he did from this F2 numbers, these numbers, he said, okay, let's add up all the rounds. All the rounds are what? This round plus this round. How much is 315 plus 108? 315 plus 108 makes 423. What is the recessive for round? Is wrinkled. Let's add up all the wrinkled. Forget about the other trait for the moment. Wrinkles are 101 plus 33. It adds up to 133. Then let's add up all the yellows, all the greens. You get the numbers, and then you calculate the ratio, and you find that 3:1 ratio is fully preserved underlying this 9:3:3:1 ratio. This ratio is called F2 phenotypic ratio, 9:3:3:1. 9:3:3:1 is the F2 phenotypic ratio for a dihybrid cross. Underlying 9:3:3:1, he discovered, I have round versus wrinkled and yellow versus green independent 3:1 ratios are fully preserved, which means when you are going from parents to the offsprings, from here to the next progeny, the two different genes, they don't interfere with the inheritance of each other. We call this law of independent assortment, which means the two pairs of alleles, independent pairs of alleles, this is one pair of allele, this is the other pair of allele, both are dictating different traits. The two pairs of alleles, they inherit independent of each other when they go from parents to offsprings. Is it clear here? So underneath 9:3:3:1, you have two hidden 3:1 ratios. This was called law of independent assortment by Mendel, where different gene pairs, they are inherited independently during gamete. And Mendel didn't use really the word gamete formation, but yes, these are the gametes being formed. So we just made these checkerboards. Abdullah, what's time now?
Sir, it's 4:40.
कितना टाइम रहता है? We started 4:20.
Yes.
So we will finish after these two slides. We'll continue tomorrow, and I would like you to read all the way up to this slide. Okay? So through this checkerboard, these are the gametes of F1 heterozygous. 4 different gametes of F1 heterozygous, and we got 9:3:3:1 phenotypic ratios. For example, this one will give you capital Y, capital Y, capital R, capital R, capital Y, capital Y, capital R, small r, capital Y, small y, capital R, capital R, capital Y, small y, capital R, small r. And when you go all the way solving here, you will say, okay, small y, capital R, and this one is this homozygous recessive, and the phenotype will be what? What will be the phenotype of this one?
Yellow and wrinkled.
What?
Green round.
Green. Green.
Wrinkled.
Wrinkled. This is for wrinkled, this is color. Okay. And when you calculate, you will find, what will be the phenotype of this one?
Yellow round.
Okay. You guys are faster than my 101 classes. What will be the phenotype of this one?
Yellow wrinkled.
Very good. Now you are not going to say anything. You remain quiet. What will be the phenotype of this one?
Green round.
Green round. Very good. So you all have gotten the concept. And what will be the phenotype of this one?
Yellow wrinkled.
Very good. You made me happy. So Mendel discovered all this in 1864, I think. And for next 40 years, Mendel's work was not at all acknowledged. People did not pay much attention, although he discovered reproducibly again and again. He proved, look, you can follow a character. You can, and from here he presented his results in the society, and he said, if you want to study the wool quality in the sheep, you need to first understand the factors which are controlling the wool quality, and then go through crossing, and that's the only way we can improve the wool quality and compete Spanish. But that's a usual scientific story. That's how we scientists live, and seldom in your lifetimes your discoveries, if you are lucky, if you are really honest and you were searching the truth, only then Allah rewards you with such big discoveries. And seldom is the case that your discoveries get acknowledgement within your lifetime. In present era, it's getting possible now, but in Mendel's time it was not possible. It was in 40 years later, in I think 1905, I believe, roughly, somebody was doing experiments with fruit flies, and they were trying to see chromosomes. Remember, at Mendel's time, this was beyond Mendel's time. 1864 was the Mendel's time. Then they had no idea about chromosomes, no idea about genes, and they had no idea about, even alleles. They had no idea about mitosis or meiosis. But in 1905, they knew what chromosomes are. Chromosomes were, they gave them name because chrom means color in Latin, and some means something which can be colored. And they were adding up a dye in the cells, and cells were showing colored structures, and they said, yeah, within the cell there is something which can be colored, and they named them chromosomes. Now they were studying different kinds of cells existing in a living organism. They crushed brain cells, the eye cells, you name different cells. And what they discovered that there were some cells which contain half cells with half the number of chromosomes, and there were cells which contained complete set of chromosomes. For example, if organism has 20 chromosomes in these cells, here in these cells it had 10, half the number of chromosomes. And these cells, they had chromosomes in pairs, complete set. For example, this is one kind of chromosome looking, the other one is like this, another one is like this, another set is, you know, like this, another set is like this. So they are 20 exist in complete set. But these ones, they were not containing in pairs. And people who were studying these mitosis and these cells, they came to conclusion that, and they could, this was a simple conclusion, they said, wow, these are the cells which contain half the number of chromosomes. These are the reproductive cells. And these ones are the normal cells of body, eyes, skin, etc., blah, blah, blah. The chromosome numbers here, they are named 2n, or we call them diploids. And when after cell division, chromosome numbers are reduced to half, which is 1n, we call them haploids. People who are studying these processes, they said, why reproductive cells have haploid number of chromosomes? Why they don't have diploids? And what is the significance of having haploids? They went through previously published literature and they discovered, wow, there was a genius who, without knowing chromosomes, without knowing mitosis, meiosis, or cell division, he postulated that a parent can only pass on half of the factor. And you know what is happening? They said, this is happening. In the meiosis, only half of the chromosomes are going, which means, let's say this is the diploid parent, this is this cell, okay? And
Sir, I have a question.
yeah,
Sir, so basically, even in the germ cells, we first replicate the DNA and then divide it in 4 cells. So
yeah.
why do we replicate? Why don't we just divide it in 2 cells directly?
I come to that point, I'll explain you, okay?
Okay.
So this is what they observed. They observed, you know, 2 chromosomes, and let's assume this is S, small s. Assume this is Mendel's heterozygous F1. Assume this, okay? S is for capital R, S is spherical and wrinkled, round and wrinkled. Clear? Everybody is on the same page?
Yes.
Now, this cell is undergoing meiosis. Like just a while ago, she was asking, whenever cell divides, before dividing it replicates its DNA. Now, all the DNA on this chromosome, it gets duplicated. And since this is exact copy, now this chromosome is this one, capital S, which is like RR, and this chromosome duplicated, small r, small r. And during meiosis, you know, meiosis has 2 phases, meiosis 1 and meiosis 2. You have studied this?
Yes.
Yeah. So in meiosis 1, these 2 similar looking chromosomes, we call them homologs. Okay? And this is what heterozygous state, ye heterozygous hai na? Capital R, small r. After duplication, the homolog chromosomes in meiosis 1, they go to opposite pole, spindle fiber. They end up in 2 different cells. And now after meiosis 2, here the sister chromatids, ये क्या चीजें थी? जब duplication होती है तो इसे हम कहते हैं sister chromatids. ये इसका sister chromatid है, ये इसके है, but together they are homolog chromosomes. Now in meiosis 2, on the spindle fibers, what happens? This sister chromatid goes to this direction, this one to this direction, and then you have two cells which have half the number of chromosomes. This is one chromosome as compared to this one, which had two chromosomes. So you end up from one diploid cell, which was 2n, you have 4 diploid haploid cells. This one, the two sister chromatids go to opposite poles. The two resultant cells, the gametes, are small r, small r. Clear? So
Yes.
that's how, what they said, okay, what Mendel was saying, actually this is what was happening, and the guy was right. We call this rediscovery of Mendel in 1905. So let me just explain you, we have still few minutes. You have a diploid cell, oh yeah, it's already drawn here. This one is, these two are homologs, and these two are homologs. Homologs means similar looking chromosomes. Exactly same. Homolog chromosomes are 100% same. They may have different alleles, but their DNA sequence is all fine. So what is a chromosome? Chromosome is like this. We already know. And the DNA. Like this. Double helical structure. Together with histone proteins. Okay? Imagine this one is with capital Y. And in a F1 heterozygous will be F1 heterozygous. For yellow will be here small y. But these are homolog chromosomes, 100% same, with few variation, few different alleles. The round ones will look like something like this. Different shape, small size maybe, etc. And here you may have capital R, and since it's F1 heterozygous, the homolog chromosome will have small r on this side. Now, she asked question, why it duplicates its DNA? It has to duplicate its DNA because without duplicating your DNA, you cannot simply pass on these chromosomes. Meiosis or mitosis fails if you don't duplicate. So after duplication, how this chromosome looks like, this will look like this, okay? All the DNA here is duplicated, and now what we have two sister chromatids. And you have capital Y, capital Y. This one is duplicated. During S phase, this one also gets duplicated, and you have small y. This is a very important concept. You have to really learn this. Now, same will be the case for this one, which is shown here, okay? Round, wrinkled, this is round, this is wrinkled. During first meiosis, they will, so this is homolog chromosome, same chromosomes which exist in pairs. So now you can see yellow and green, they align on the spindle fiber, yellow and green. Only homologs pair It will never be this chromosome pairing up with this and this one with this. Only homologs pair. The chromosome pair which contains round and wrinkled, they also pair. And on meiosis 1, what happens? They go to opposite poles. Okay? After going to opposite poles, we will have two cells after meiosis 1. And this one will be capital Y, the chromosome, this one, or, and, sorry, round, its corresponding chromosomes. This one can be small y homolog, and which is wrinkled. But it can also go like this. Inheritance, remember, law of independent assortment was that this can go during gametogenesis with this one, which means this is also possible. And on this side, this is also possible. This is law of independent assortment. And then in F2, when you go, in meiosis 2, now these sister chromatids, which was this one or this one, they go to opposite poles. And you get, round, green, round, yellow, wrinkled, green, gametes. Okay? So this is a very important concept which you should today go and read, because tomorrow we are going to pick exactly from there. We are going to pick it, and we'll go to very exciting work of T.H. Morgan, which is Abdullah's favorite guy. Abdullah is your T.H. Morgan of Pakistan. Okay. So if you will understand Mendel's two laws and these concepts of inheritance during meiosis and independent assortment, you will have real fun going to Morgan's experiments, which really challenged Mendelian laws. And the question Morgan threw was, what will happen if I have both yellow and round instead of on different chromosomes? What if yellow is here, round is on if they're on the same chromosome? Mendel could not understand, because at times of Mendel there was no concept of chromosomes. There was no association between genes and chromosomes. So whatever Mendel discovered, Mendel was extremely intelligent but extremely lucky, because whatever Mendel discovered, all the genes, traits he studied, all the traits he studied, they fell on seven independent chromosomes. The pea plant has 7 pairs of chromosomes, and Mendel studied 7 traits. Each trait was on 7 independent chromosomes. And that's why Mendel was getting so beautiful law of independent assortment. Always, no matter whichever trait, a dihybrid cross he was setting up. In my personal humble opinion, this is not possible unless nature does not want to reveal its secrets. This is only possible once in a century, when nature wants to reveal some secrets, without knowing, the chromosomes, the genes, chromosome pairs, you discover laws which holds true today and even beyond. They will be holding true till the final day of this universe. So if you understand this, we are going to talk about what will happen if you have genes existing on same chromosome. Will you get 9:3:3:1 ratio or gametes like this? 1/4, 25% each, or ratio will change. That is what we'll talk tomorrow. Very exciting lecture tomorrow. Any question?
Sir, I have a question. Sir, like we just talked that this yellow, all the traits are contained on these chromosomes. So obviously each chromosome does not only contain like one trait. So there are many other characteristics as well. So don't they get affected when these pairs form?
No, when you are, your question is that there are many other genes on the same chromosome. So
Exactly.
why don't they impact yellow or round or spherical? Because you as a Mendel are only concerned with yellow round or yellow round, wrinkle, tallness, dwarfness. So visibly you are only picking up plants which are giving you phenotypes which you included in when you started an experiment. Of course, there are maybe 15,000 genes in pea plant, maybe 20,000. I don't recall the exact number. But you are choosing only 2 traits. That's why whatever the others are doing, they are of no concern, and they don't show you any phenotype because you don't look at their phenotype at all. Did I answer your question?
Yes, sir. Thank you.
Any other question?